Can everyone hear me? Yeah?
Yes.
Cool. So I'm going to talk to you today about microbe-mediated host defense and how it drives the evolution of reduced pathogen virulence. So what is microbe-mediated defense? As you all know, your microbiota is pretty largely responsible for your health, and there are a lot of defensive microbes, that I call them, in your microbiota that are largely responsible for that. And this is what I'm looking at. Sorry, I'm really nervous. It's a really big room. Okay. Yeah, if you laugh, that makes me feel better. Okay, cool. So, yeah. So what I wanted to know is basically how these defensive microbes interact with pathogens, and there's quite a lot of defensive microbes known across the plants and animal kingdoms, and they are largely responsible for protecting hosts against parasites, parasitoids and pathogens. I'm going to just use the term pathogen, and they've been quite a key sort of point of interest for alternative candidates for disease control. So, for example, phage therapy in the treatment of human microbiomes and things like that. So yeah, I wanted to know what effect these have on the evolution of pathogen virulence, because we know that pathogens can evolve with other sort of challenges to infections. So I know that their virulence has altered in response to immune-priming vaccines, and also the virulence is predicted to evolve in response to competing pathogens and other challenges like that. So the fact that defensive microbes can have quite a large implication or impact on pathogen fitness means that they're also likely to have strong selection pressures for pathogen virulence. So I came up with two mutually exclusive but equally plausible relationships or hypotheses of how pathogen virulence could link with the the resistance to defensive microbes. And I don't have a pointer, I don't think, but on the left is positive correlation. So this is quite simply when the virulence of pathogen correlates positively with its resistance to a defensive microbe. This might be the case if the resistance to a defensive microbe is merely having a higher, faster growth rate or something to counteract this defense. And in that case, a defensive microbe would select for increased virulence. And on the right is the case of trade-off, basically where this may happen when the resistance trait to a defensive microbe is costly. So, for example, if a defensive microbe produces antibiotics and the pathogen becomes resistant to that, and that's actually costly to its growth rate, so it has a lower growth rate and so lower virulence. And so in this case, the defensive microbe would actually select for reduced virulence.
Nice water.
Oh, thanks. How do you use it?
Wrong way.
Don't point it at your eye.
Well, now I can't see my presentation. Okay, so I looked at this question using the C-Norhabditis elegans system, which is a neat model system because it sort of eats bacteria for a living. So, it's been used to study microbial pathogenesis. So I used Staphylococcus aureus as the pathogen, and this particular one was isolated from a virulent infection. And I used Enterococcus faecalis as a sort of commensal gut microbe, as this one was isolated from that sort of infection. And so what you can see from here is all my experiments are done within 24 hours because of experimental evolution purposes. So on the y-axis is the percentage of hosts that are dead after 24 hours of exposure to the bacteria. As you can see in the centre, S. aureus, the pathogen, can kill up to 30% of the worms in 24 hours, so it's pretty virulent, whilst Enterococcus is really barely virulent at all. And under the co-colonization treatment, the Enterococcus reduces the virulence of staph by quite a significant amount. So in this case, you could consider it as a mimic of a defensive microbe that you'd see in nature. And so looking at the bacterial accumulation, you can see here that basically the fitness, so the number of cells in the worm of Staphylococcus, the pathogen, after 24 hours is significantly reduced in the presence of the defensive microbe. And we know from previous experiments that this defensive microbe produces superoxide, and that really harms the pathogen. So that's almost certainly 100% why we have this reduction in fitness of the pathogen. What's really interesting, though, is that if you look at the fitness of the defensive microbe, you can actually see that it increases in fitness in the co-colonization treatment. So it's actually benefiting from the presence of this pathogen. So you actually have quite a strong antagonistic interaction here, more like a host parasite, if that's what you recognise. So to perform the evolution experiment, I basically had two treatments: a co-evolution treatment where the pathogen and the defensive microbe are co-passage, and a single evolution treatment where the pathogen is passaged on its own. So all of the treatments are started from a single ancestral population, and the worms are not evolving in this experiment, just to let you know. And basically, for each generation, passage generation, I exposed them for 24 hours to about 1,000 worms per replicate. I did about 5 replicates for each. And after this time, the guts from 10 worms were placed onto selected media, and 10 colonies of each bacteria were picked. And these were grown up independently and used to start the next passage. So for the next slides, I'm going to just use this worm as showing you basically that it's an in vivo experiment. And when it's in vitro, I'll use the tube just to make it really, really clear what's what. So what happened to the virulence? Basically, I found that co-evolution with a defensive microbe selected for significantly less virulent pathogens. And so this is this treatment here, co-evolution, and it's significantly less virulent than the ancestor, but also the single evolution control, which is the key treatment to compare it to. So I kind of wanted to know why. That's really interesting. And so naturally, I correlated it against growth rate, as many people assume that it's to correlate against growth rate, to increase with growth rate. And indeed, I find positive correlation, and just as you found the co-evolution treatment reduced in virulence, it also was reduced in growth rate. So this is cool. So I was kind of thinking, what might be linking the virulence with the growth rate, and what trait might this be to make it beneficial in a social context? Oh yeah, before that, sorry. So I wanted, just to show that this is an adaptive trait. So basically, I put the different Staphylococcus treatments, so the ancestors of co-evolution and single evolution, against a common defensive microbe. So this is the ancestral defensive microbe, and you can see that the fitness of the co-evolved one is significantly higher. So whatever's going on is actually adaptive, it's beneficial. So now we can look at why. So the link between growth rate and virulence in a social context led me to think about siderophores. Siderophores are molecules that are produced by microbes to bind iron in the environment and uptake it, and iron is really important for their growth. Okay. So because iron is important for their growth, siderophores inherently link growth rate to virulence. What's really important here is that siderophore production is also a social trait. So just as you might produce siderophores into your environment to take up iron, another neighbouring microbe that might be of a completely different species can also take up that iron, which means that basically you're being exploited. So current sort of theory, skin selection theory, would propose that if this species here is unrelated to you, either a strain or a species, then this producer strain or species would be selected to produce fewer of these siderophore public goods. That's what they are, public goods. What I sort of proposed was that this selection would be even stronger if this species is also harming the producer species. And so basically, I tested this, whether siderophore production was the trait, and it was, which is really lucky. So basically, I find that the co-evolution treatment, there we go, in the middle, produces significantly fewer siderophores than the ancestor or the single evolution control. So I thought that's great. So want to know whether this was responsible for the virulence reduction that we saw, and it is. So you can see here that the lower producers of siderophore are also the less virulent in the co-evolution, and so between and within the treatments. Okay, so I wanted to see from this whether this was actually adaptive in the social context, so whether it was affecting its interaction with the defensive microbe. And so let me talk you through this graph. So this line here is the fitness of the defensive microbe without the pathogen. So if you remember, we have this increase in fitness when you put it with the ancestor because it's got this sort of benefit. What you see is that this fitness benefit remains with single evolution control but disappears with the coevolution treatment. So that's indicative of it being responsible for that. But I want to know explicitly whether reduced Siderophore production was actually responsible for this benefit to the defensive microbe completely disappearing. So what I did was basically, if you add surplus iron to the environment, Siderophore production necessarily downregulates, doesn't need it so much, so you get this trait disappearing. And so you can measure what happens in the absence of changes in Siderophore production. So this is what I did. The control is what I just showed you before, so the coevolution treatment benefits Enterococcus significantly less, the defensive microbe. And on the right is where I add surplus iron, and this treatment, this difference, it disappears. And this is basically driven by the fact that the Enterococcus regains its fitness. So the defensive microbe basically is no longer benefited with the co-evolved pathogen. Yeah, so basically the co-evolved pathogen evolved reduced Siderophore production in order to reduce the fitness of the defensive microbe, and in turn that's how it's adapted. So to conclude, I found that defensive microbes can shape pathogen virulence evolution, and that in this case, Staphylococcus pathogen became less virulent as a byproduct of adapting to the defensive microbe, and this was via Siderophore public goods. And this supports the second model that I presented at the start, the trade-off model, which could be applied to other systems if that exists. And finally, that defensive microbes could therefore not only protect their host in the short term, but they could also select for less virulent pathogens in the long term, which is pretty cool. So I'd just like to thank my sources of funding, St John's College, Oxford University, and my supervisor, Kayla King, and my lab mates for all of their support. Thank you.
Time for a question.
Yeah.
You had one cobalt data point that was off a little bit.
Yeah.
On the Siderophore.
Yeah.
Did it not have the same kind of fitness consequences? Or was
It did. Everything else with all of that data was consistent, so I'm wondering if that one data point was actually like an anomaly. So it could have been, but the other data surrounding it doesn't suggest that. But so it's possible that it's an anomaly, but without that, the R-squared is like 90 something, which is great. So. But yeah, maybe I'll repeat it, who knows.
Quick one.